Hello, this is a histology tutorial on glands presented by the SchrDbb with reference Atlas of Histology and Functional Clinical Rotation by Dongmei Cui. So, we're going to start here with glands. So, to know glands, we are going to start with the introduction and the key points on glands. So, for the introduction of the glands, you need to know that generally glands are going to be divided based on their function and based on their so we have different division of glands. But to point out the fact is that the glands that you're going to mainly see here are going to be glands of the exocrine glands, because based on the functions, glands are going to be divided into endocrine and exocrine glands. An endocrine gland is a gland that directly secretes its product into the bloodstream or directly into the interstitial fluid. While an exocrine gland is a gland that secretes mostly into a duct. Is it clear? So those are the main two divisions of glands. So glands can either be divided. So that's the first division of gland, that is based on endocrine and exocrine gland. An endocrine gland is one that secretes its product directly into the blood vessel or directly into the interstitial fluid. And then an exocrine gland is the one that secretes directly into the ducts. Now sometimes endocrine gland can be said to be, so sometimes we can use another word of gland being merocrine glands, paracrine glands, and so sometimes we have other types of glands like paracrine glands. Now a paracrine gland is a gland that is secreting its product directly into the interstitial fluid. You see the definition I've gave initially to endocrine gland. So usually some textbook can say that endocrine glands secretes product directly into the bloodstream, while paracrine gland secrete product in the interstitial fluid in order to make sure that the target cell is only a local cell. While for endocrine gland, it secretes into the blood vessel, and the target cell is a systemic cell, the cells which is found at a very distant area. Now we have also autocrine glands. Autocrine glands are glands that are going to produce product that are going to stimulate itself. Is it clear? But those is only based on the function. But what we're going to mostly visualize here are going to be exocrine glands. So now on exocrine glands, you need to point out the fact that we have the classification of exocrine glands based on the product they secrete. Now it can be a serous gland if it produces watery product and proteins. It can be a mucous gland if it produces mucus. It can be a mixed gland, that's seromucous, if it produces both serous product and mucous product. So those are the three main types of glands based on the product they secrete. Sometimes you may add sebaceous gland as being one based on the product they secrete, because sebaceous gland produces purely lipid substances. Now the next one that you have to point out here are the exocrine gland classified by the mechanism of secretions. So the exocrine gland classified by mechanism of secretions, we have the merocrine gland, and the merocrine gland when they secrete, there is no loss of part of the cell. Secretion is by pure exocytosis. The second one is the apocrine gland. So for the apocrine gland, you have the loss of the upper part of, when it is secreting its product, there is loss of the upper part of the cell. Is it clear? So in apocrine gland, an example of location where you have apocrine gland is in the case of the mammary gland. Example of location where you have merocrine gland is in the case of the sweat gland or even the salivary gland. Now the last one is the holocrine gland. For the holocrine gland, we have the secretion of the product is when you is with the loss of the whole cell. So in order for you to secrete the product of the gland, the whole cell is going to be lost. Example of this is going to be sebaceous gland. Now, the next thing to visualize is the classification of the exocrine gland based on a morphology. So morphology is really what brings us under the subject matter, which is histology. That is, morphology is the main thing that brings us here. So in order for you to actually know the histology of an exocrine gland, you must know the classification based on morphology. It may seem complicated, but it's actually a little simple. Now, we have to know that what the morphology, you can either have unicellular gland or you have multicellular gland. And unicellular and multicellular gland may have again order underneath it. So it's just like for stratified squamous epithelium and simple squamous epithelium. For simple squamous epithelium only contain one cell layer thick, while for stratified squamous epithelium it has multiple cell layer thick. Now this is unicellular gland. Unicellular gland only one cell that it is inside the gland. Is it clear? It is not one cell layer. No, it is one cell which is the gland. Is it clear? Unicellular gland, one cell is the gland. Now we have multicellular gland. In multicellular gland, what is there? In multicellular gland, it is classified now into two. We have simple glands and we have compound glands. So multicellular glands is now made up of many cells which is going to be producing the gland. So it can be either a simple gland or a compound gland. Now, simple gland can be divided into simple tubular. So when it is a simple gland, so a simple gland is usually, what is the specific characteristic of a simple gland? A simple gland is unbranched, while a compound gland is branched. That is the main difference between simple and compound. Now, simple gland, which is unbranched, for you to say that it is multicellular, it must be either simple or compound. So simple is unbranched, while compound is branched. Now, simple and compound can also be tubular or acinar. Tubular means that what? So when I have a simple tubular gland, it means that it is an unbranched and it is looking like a tube. So the gland is just like a tube. We take your test tube, you know the morphology of a test tube, right? So you take your test tube, just like the inferior portion of the test tube, like that. That is how it's going to be looking like. While for an acinar gland is just resemble like an alveoli. You know that we also have, the round bottom flask in chemistry. So the round bottom flask, as you hear the name, is just having a round bottom. It has that same morphology in acinar glands. So it is, when I say simple acinar, it means that it is simple and it is unbranched, and it's having the shape of a round bottom flask. Is it clear? Now, sometimes we have also another form, that is a simple coiled acinar and tubular gland. It is just having a tube. It is unbranched, it is tubular, and then it is coiled. That's simple as that. So those are the two main. Those are the different simple gland that you can have. The other ones, sometimes, though it is said that simple is unbranched, you may have a form of simple branch that we're going to see in the later characteristic. So in general, the different simple glands that you have simple tubular gland, simple branch tubular glands, you have simple coil tubular glands, you have simple acinar glands, you have simple branch acinar glands, you have compound tubular glands, you have compound acinar gland, and you have compound tubuloacinar. This one contains both a tube and a round butterfly appearance. We're going to see it in the picture below. Now, the next thing is a duct system. So when you speak about the exocrine gland, you have to make sure that you also know the ducts of the endocrine gland. Now, the duct system of the endocrine gland, so after having the acini and the tubules, the acini and the tubules are usually arranged in lobules, and those lobules are going to be secreting into ductules. And one particular acini is going to secrete into a ductule. A ductule is a small duct. One particular acini or a group of a gland is going to secrete in a ductule. And now when all those glands which are located in a lobule are going to secrete, the lobule now is going to be connected by a duct. So these are now what is going to remove the secretory product of the lobule is going to be small intra. So in between the lobule we have intralobular ducts. Okay. So it may also be referred as intercalated ducts. So we have also small intralobular. After draining, is going to drain now into the larger intralobular ducts, and then it's going to secrete now into the striated ducts. All that we're going to visualize later. So now let's see the specific image now of the exocrine glands. So the specific image, you need to know that this is an example of a parotid gland. So the parotid gland is an example of a serous gland. So in serous gland, how does it resemble? So this is a small image. Inside here is a lumen. Here is a glandular cells just above here. Those cells are serous cells. So here is an example of a mucous gland. A mucous gland, here inside is the lumen. You have the lumen, and here above here, so laterally here, you have the glandular cells. And usually these glandular cells are called mucous cells. You see, the glandular cells in serous cells are actually having a different appearance than glandular cells in mucous cells. I'm going to show you those differences below. Now, mixed gland. You see, the mixed gland has both a serous gland and also has a mucous gland with a lumen. So you cannot actually visualize well the lumen. In this case, you may have this is the lumen here. So this is the lumen here. So you see that the serous gland are usually producing a demilune. So they have a lunar appearance. You see the lunar appearance also here. So that those are for the serous gland. They are producing a lunar appearance. That's why it's called the demilune. Now the next thing is for the sebaceous gland. For the sebaceous gland, this is how the appearance now of the sebaceous gland on the microscope. So for sebaceous, see, these are the lipid secretory gland. So they are whitish in character, basically. So let's continue. So you have and I've forgotten to also say that in the mixed gland, you have a demilune of the serous gland, and you also have the central mucous cells here. These are the serous cells, and this is central mucous cells. Now, the next point that I want to show you now is going to be the electron microscopic view of a serous acinus. So this is a serous cell. This is serous cell here. So for how does a serous cell resemble? So see, serous cell has rough endoplasmic reticulum, not much. It has the nucleus. It has some secretory granules inside. It has these are some secretory granules inside this like that. So secretory granules, as you can see. secretory granules. So that is for a serous cell. Now, this is the microscopic view now of electron microscopic view now in mucous cells. So you see the difference between, so this is the lumen, so this is one serous cell here, this is another serous cell here, the nucleus of this serous cell, this is the granule here, which are going to be secreted. Now for a mucous cell, you can visualize here the nucleus of this particular mucous cell and its nucleolus. But you see that with the nucleus cell, at the base layer you have a nucleus, and the apicalmost layer you have many secretory granules in the mucous cells compared to the serous cells. The serous cells do not have that much secretory granules. So their secretory granule is not that much compared to the mucous cell. See, the mucous cell has many secretory granules inside like that. So that is why in the image that we've seen on the, you can just pause and go back behind. You see that the image that we've seen just above there was having a whitish apical appearance at the level of the mucous gland. So you see many whitish appearance inside the mucous gland, and there inside is the lumen of the particular mucous acinus. Now let's visualize now the glands based on their morphology. The first was the visualization of the glands based on their function. Now this is a visualization based on their morphology. Unicellular glands, we said that unicellular glands only consisting of a single layer of cells. Example of unicellular gland is a goblet cells. You see, this is the microscopic appearance of goblet cells. When the goblet cells secrete, they directly secrete in the lumen where it's going to act. It does not first pass into ducts, or it does not combine with any product of another gland. So it directly secretes into the lumen where it's going to have its mucous function and protecting the gastric lumen, so the gastric mucosa. So in this case, this is a microscopic view, the electron microscopic view. You see, this is actually like a mucous. This is actually a mucous gland. So this has a typical appearance of the mucous cells that we have seen before there. So goblet cells is one mucous cell, which is a gland itself. Is it clear? But in case where you have multiple mucous cells together to produce a gland, you have now a multicellular mucous gland. Is it clear? So that is it. So here these are the microvilli of the enterocyte, because they are also simple columnar epithelium just close to it. So here is a goblet cell. So this goblet cell is having secretory product. This is why the goblet cell in the light microscope is going to be whitish on the hematoxylin and eosin stain. It's going to be whitish here, because there are many secretory granules which are consisting of mucous material. Then the nucleus is going to be located underneath, and then you have some rough endoplasmic reticulum, the Golgi complex, because it's used in the production of this much secretory granule, and you can have also some smooth endoplasmic reticulum inside here. Now the next thing is that we have multicellular glands. So for multicellular gland, it can either be a simple tubular. This is a schematic representation of a simple tubular. Simple tubular, as I said, the base is going to be like a test tube, and then it's going to be having a such. This is a simple tubular gland. So the schematic representation. Now this is the histologic representation of a simple tubular gland. So for simple tubular gland histologic representation, what do we show? So this, let's say this is an example here. So this is an example of simple tubular here. So you can see actually the tube here. You see the tube here. So this tube here is, you see here, these are the cells involved in the simple tubular glands. These are the cells involved here as such. Now this is based on the morphology. Now to know the function of this gland, you may now increase the magnification. The function may be serous function, and we have seen the morphology of the serous gland just above there. So you can just pause and go back and see the morphology of the serous gland. we said that the serous gland on the hematoxylin stain is going to be much more pink, while the mucous gland on the hematoxylin stain is going to be much more whitish. You see, and the apical muscle layer is going to be whitish. For the mid gland, it has demilune. It has a demilune of the serous gland, and then the midpoint is going to be mucous gland, and then it has a lateral also another demilune of the serous gland. And then you also have the last one, which is a sebaceous gland, where it is all whitish around. There is no apical part which is whitish, so more than the base, it is whitish all around for sebaceous gland. So that is when you want to find the function, but only the morphology. This one here is like a tube. So since it's just resemble like a tube like that, and then the cells are inside, this is why it is called a simple tubular gland. Now, for a simple branch gland, this is diagrammatic representation of a simple branch gland. So this is a cell, so the epithelial cell. You see, there is only one pit which is connecting them. So this is why it is simple. So simple is because there is only one pit that is connecting them. So this is going to be a tube and a tube connected as such like that. So this is a simple branch tubula. So now this is the histological representation of a simple branch tubula, where you have this and this. So here it is located in the gastric pit, also in the stomach. So this is a gland located in the stomach. So this gland is located in the large intestine. Now the next thing is a simple coil tubular gland. So this is a diagrammatic representation of a simple coil tubular gland, where what happens, so you have the excretory ducts and you have the tube. So the tube, now you see that the tubular part is going to be slightly coiled. So you see, tubular part is going to be slightly coiled as such here. That's why it's said to be simple coiled tubular gland. So here you are going to have the excretory duct, and then the secretory part now is going to be in a coiled portion. So this is now a diagrammatic representation. You see here that the cell here is a bit coiled, because now you see the section has been transverse in this direction. So this is why the coil is not easily seen, but you see actually the loop is actually slightly coiled like that. This loop is slightly coiled. This loop is also slightly coiled. So here is the lumen, and here are the secretory cells actually close like that. Also here is slightly coiled. Here is the lumen, and here are the secretory cells. So the next thing is the simple acinar gland. So you see that the acina, as I've said, is just like a round bottom flask. At the beneath part is now like a brown bottom flask. Is it clear? So this is, you have the excretory duct here, and the secretory gland are producing a round bottom flask, not a tube. So that's why simple acina. So this is an example here like that. You have a simple branch. So this one is simple branch. It's not actually acinus. Is it clear? So it is a simple branch acinus. Here it can be also a simple branch acinus, but here like this, it is much more of a simple acinus. So is it clear? A simple acinar gland. So here is simple branch acinar gland. Here is simple branch acinar gland. Here is much more of a simple acinar gland. Here around is the secretory cells, and here is going to be connected by the excretory duct into the lumen. Is it clear? So it's also going for this part here. So we have simple acinar glands, much more located in the lithry glands inside the submucosa of the male urethra. That's an example. Now the simple branch acinar gland. For the simple branch acinar gland, you see that there's only one excretory duct connecting them, and then many of them are now having acinus like that. So these are acina. So this is an example of a simple branch acinar gland. So here is the excretory duct. Here is the first acinus. Here is the second acinus. Here, simple branch acinar gland. So this diagram has been taken on the skin. Now the next one is the compound tubular gland. So for compound, what happens is that each of them has a excretory duct. Are you seeing? Each of them has an excretory duct, a particular excretory duct. For the first simple branch, when it is simple branch, all of them have a common excretory duct, and then the acinus is only joining, is only the acinus is the one that is branch. It is simple because it only has only one duct, and then the acinus are branch. Now it becomes compound when there are multiple excretory ducts. So in this case, you have 3 excretory ducts which is connected to 3 branch tubules. So that's why it's said to be compound tubular glands. So it is different excretory duct and those are branch. So that is a compound tubular gland. It can be seen here. This is seen in the duodenum, the Brunner's gland of the duodenum that produces the intestinal fluids. That produces the intestinal fluids. Then the next one is a compound acinar gland. For the compound acinar gland, it has multiple excretory ducts, and then each of those multiple excretory ducts has a branch. So the compound acinar gland, it has multiple excretory ducts, and each of it has a branch acinus. So the acinus here is branch, acinus here is branch, acinus here is branch. So that is why it is now said to be a compound acinar gland. So example of compound acinar gland, you have it inside the pancreas, exocrine gland. The next is compound tubuloacinar gland. So in compound tubuloacinar gland, you have still multiple excretory ducts, but now some are going to be branch tubules and the others are going to be branch acinus, as you can see here. So you have branch tubule, branch acinus in compound tubuloacinar. So here is an example of a compound tubuloacinar in submandibular gland. And submandibular gland, you also remember that it was also a mixed gland, that is, it has a serous part and it has a mucous part. The serous part produces the demilune and the mucous part is at the center and it is whitish. The whitish part is because you have the apical, it's not whitish all around. So in order to know that it is a mucous cell, you have the nucleus at the base, and then it is whitish at the top. Is it clear? Why in case of a sebaceous gland, the nucleus is in the center and it is whitish all around? Is it clear? That's the difference between a mucous gland and a sebaceous gland. So we continue now with the different parts. So we continue with the next part of the tutorial. Thanks for your kind attention.